Hi guys, welcome to Draw My Research, presented to you by UCLA's Breaking Back team. I'm pretty sure you've heard of bacteria before. What comes to your mind when you think of bacteria? Pathogens? Disease? Dirt? Well, not all bacteria are harmful to us, and there are actually a lot of benefits we can get from bacteria. They help to promote plant growth, produce antibiotics, and help to produce renewable energy. These functions give bacteria many medical, agricultural, and industrial uses.
We began our research by collecting soil samples from Sedgwick Reserve, California. Sedgwick Reserve is unique because it contains patches of serpentine soil. Serpentine soil differs from regular soil as it has a high magnesium to calcium ratio and low nitrogen levels. This harsh environment makes it difficult for plants to grow as plants need these nutrients to grow well. We collected 4 soil samples, 2 from grassland and 2 from serpentine soil. We cultivated 24 bacterial isolates from one of the serpentine soil samples. Some of the isolates were white, some of them are pink, and some of them are yellow. Based on what we know about serpentine soil, our group hypothesized that microbes found in the serpentine soil could fix nitrogen, produce antibiotics, and promote plant growth through a variety of pathways. These characteristics which contribute to plant survivability in serpentine soil.
What is nitrogen fixation? How does bacteria play a role in nitrogen fixation? Nitrogen fixation is a process that converts atmospheric nitrogen into ammonia in the soil that plants can absorb. Some microbes live in the roots of plants and perform nitrogen fixation, forming a beneficial symbiotic relationship for the plant. Microbes with the ability would be particularly beneficial for plants living in low nitrogen serpentine soil. So, we decided to test our soil sample for the presence of nitrogen fixation bacteria.
To test for isolates' ability to fix nitrogen, we first streak our isolates onto N2- bat media, which lacks usable nitrogen so that only bacteria that can produce ammonia on their own are able to survive. Then, we inoculated the bacteria that can grow on N2- bat plates into different types of media such as JMV, DF, and N2- free media to further test for their nitrogen fixing ability. If isolates can grow in these media, then growth can be observed at the top part of the media in the tube. So far, we have 6 bacteria that demonstrated growth in these 3 types of tubes.
Are you familiar with antibiotics? Did you know that some antibiotics come from soil bacteria? Some soil bacteria secrete antibiotics to kill other bacteria to reduce competition and get more nutrients from the surroundings. Even us science majors didn't know that before we took this class. Most of us have been prescribed antibiotics at some point, so I guess we are no strangers to antibiotics. Some bacteria have developed antibiotic resistance, meaning antibiotics are no longer effective in treating diseases caused by those bacteria. This is why some scientists are hoping they can find new antibiotics produced by soil bacteria to treat diseases.
To test if our bacteria can produce antibiotics against other microbes, we grew our isolates with other bacterial strains on the same plate. If an isolate is capable of producing antibiotics, it will have a clear circular zone around it, indicating that it inhibits the growth of other microbes around it by producing antibiotic against them. A bigger zone of clearing means that the bacteria produces antibiotic at a higher concentration. So far, we tested 20 of our isolates, but could not find any zone of clearing.
Some bacteria even provide functions that directly promote plant growth. Plant growth promoting bacteria are particularly useful because they can be harnessed for agriculture. One benefit of using plant growth promoting bacteria is the reduction of fertilizer use, resulting in less runoff pollution and fertilizer manufacturing. One method that bacteria use to promote plant growth is solubilizing phosphate. A lot of the phosphate present in soil is insoluble. By converting insoluble phosphate into a soluble form, the bacteria allows for more efficient uptake of phosphate by plant roots. Another method is IAA production. IAA is a growth hormone that directly enhances plant growth. One more method we will talk about is siderophore production. Siderophores convert insoluble iron in the soil to soluble iron so that plants can absorb it for healthy growth. We tested for the presence of bacteria that can perform these 3 plant growth promoting functions with different assays that we will talk about next.
To test if our bacteria are capable of solubilizing phosphate, we use a PVK plate. This type of plate has a phosphate source called TCP, which has very low solubility. We grew our isolates on PVK plates and incubated them for 1 week. If the TCP was successfully solubilized by the bacteria, a clear halo will form around the bacteria after incubation. This demonstrates the bacteria's ability to solubilize phosphate. So far, we had good numbers. So far, several of our isolates showed clear zones.
To test for IAA production, our isolates were first grown in media with high tryptophan concentration. Tryptophan is an amino acid that acts as a precursor for IAA production, meaning that plants convert tryptophan to IAA through the biosynthesis pathway. Grown isolates will be mixed with the DMCA reagent on a filter paper. If IAA is present, the DMCA reagent will turn the filter paper a purple color. So far, we tested 22 of our isolates, but haven't found any indication of IAA production from our isolates.
To test if our isolates can produce siderophores, we use a special plate called a cast plate, which is very useful to test siderophore production as the production of siderophore can be monitored via color change. The plate originally has a rich blue color due to the iron in the plate, but the presence of siderophore yields a color change from blue to orange. We have grown 22 of our isolates on cast. Only 1 isolate so far has produced an orange halo, indicating siderophore production.
Our research may result in possible benefits that could impact global agriculture and healthcare. For example, if we find plant growth promoting bacteria, these bacteria can be directly used by farmers to enhance cultivation of crops with the use of fertilizer. This would decrease agriculture pollution and waste. Furthermore, if we find bacteria that produce novel antibiotics, these new antibiotics can be used in pharmaceuticals and healthcare. We could potentially find antibiotics that are less easily resisted by pathogenic bacteria.
Throughout our research, we ran into many challenges. One of the biggest issues we ran into was time management. Within an 8-week span, we formulated our hypotheses, planned our experimental approach, and executed our experiment. We constantly worried about running out of time, making mistakes in our experiments, and staying within our budget. One thing we learned is that communication is key to working together effectively. As college students, all of us had to work around our different schedules, and on top of that, manage our other classes, MIMG 101. Overall, we were able to overcome our challenges and complete our experiment and had a blast along the way.
Thanks for watching Draw on My Research by Breaking Back. We hope you enjoyed our presentation and learned something new about the awesome world of microbiology.